you've probably just got a little notification there. And on campus, obviously, all the research that we do is related to genomics across lots of different areas. And Hilary this evening is going to talk about the human microbiome and the research happening on campus around that. A little rundown of this evening's session then. Hilary is going to give us a talk for about half an hour, and as he talks, you're welcome to pop questions in the chat box or just chip in if you have any. After that, there'll be time for a Q&A with Hilary. And then Fran and I will lead a sort of discussion on teaching the microbiome in the classroom, some resources that you can use and sort of other bits like that. We also use a Padlet as we do that, so that you can also share activities and ideas that you might have as well with each other, because we find that is usually quite useful. Okay, that is all from me, so I'll stop sharing and I will hand over to Hilary.
Okay, hello everybody. Can you see my screen?
Yeah, perfect.
Okay, hi. So thanks for the introduction, Em. So as she said, my name is Hilary. I work in the Sanger Institute, and I'm going to talk to you today about the human microbiome. So this is based on a talk I gave to students a few weeks ago. So do feel free to interrupt me during the talk with questions or any queries. Sometimes it's nice to know that there's other people listening when you're doing these Zoom calls. Okay, so we'll start. So to start the talk, there was just a simple question of, have you heard of the microbiome, and what comes to mind when you hear the word microbiome? And I think generally the students, most of them had heard of the microbiome and had some understanding of what it was. So to answer that question, I started with this one to get people's attention. So the question is, what's in your poo? And this is a really important question from my perspective, because this is the work I do. And actually, most of it is water, and the rest of it is solid material. But there's lots of microbial cells. So this was an estimate that some people did a few years ago. So they estimated there was 10 to the power of 11 bacterial cells every time you go to the loo, and bacteria making up the largest constituent of the microbial cells. And there was also a high proportion of archaea and viruses and other microorganisms, fungi as well. And also a lot of colonocytes, which is your epithelial cells. So these are your epithelial cells in your gut, which we naturally shed every few days, and these pass out in fecal matter as well. And so all together, all these microbial components comprise the microbiome or the microbiota. And there are different definitions for these two terms. The microbiota is the collection of microorganisms, so bacteria or viruses, like I showed you on the last slide, living in a particular environment. And the microbiome is the genomes of the microbiota. But people use the terms interchangeably, and you're going to find I use them interchangeably as well during the talk as well. But that is their strict definitions. So as humans, we are colonized by a very diverse intestinal microbiota. And so as you move down from the mouth, down the GI tract into the stomach, we get increasing abundances and diversity of different microorganisms. And it's down in the colon, or the large intestine, where we see the greatest abundances and the greatest diversity. And even within the colon, we see different communities as well. So in the lumen, so this is the part in the middle of your large intestine, we find some bacteria prefer to be in here, and other bacteria prefer to be associated with the mucosa. So this is a layer of cells, like a gel, that sits on top of the epithelial cells. So it's quite remarkable. We have one layer of epithelial cells that separates us from the insides of our bodies to the lumen. And as a result of this, the immune system is highly active in the gut because we are ingesting foreign substances such as food and other bacteria that could potentially make us sick. And as a result of this, our immune system is highly switched on, and it interacts with our intestinal microbiota, and they help train our immune system, and it learns to tell pathogenic bacteria, so bad bacteria in the simplest terms, from commensal bacteria or good bacteria. And so really the point here is that, you know, you are a complex ecosystem colonized by hundreds of different bacterial species. And I think using the idea of a rainforest gets this point across very well, that you have all these different species interacting with each other. And we know that bacteria in the gut interact with all these different food webs. So the one bacteria eats a certain nutrient source and produces a certain metabolite, which is food then for another source. So we have bacteria interacting with each other. We have bacteria interacting with us as well. So the bacteria in the gut, and I'm going to focus on the bacteria for this talk, because we know they make up the greatest biomass, and in terms of research to date, they play a big part in human health and disease. But just as an aside, there's a lot of interest in the other microorganisms in the gut, especially like bacteriophages. So these are viruses that infect bacteria. So we as humans have viruses that infect us, and bacteria have the same as well. So the bacteria that are within our gut, these are not transient passerbys. They're highly adapted and are highly evolved to living in our gut. And so what I'm showing you here is it's the phylogenetic tree. So this is done by taking the DNA sequences from different microorganisms and comparing them to each other. And the more similar the DNA of an organism is, the more closely they cluster together. And so this is quite a complicated one, but it shows the tree of life. So down here we have the eukaryotes with humans, and we're just like a tiny branch down on the tree of life. But the vast majority of the diversity is bacterial. And these four groups I've highlighted here, the proteobacteria, the bacteroidetas, the actinobacteria, and the firmicutes. These are the four main groups that are found within the human gut. So this is at a high level classification at the phylum level. As you go down, you get lots of different families and lots of different species. But if you look across different human populations around the world, you find these four main phyla. So this indicates that they are highly evolved to living in the gut. The bacteria in your gut, you don't find them in the environment because they cannot survive there. So these bacteria, the majority of them are anaerobic, so it means they cannot survive in the presence of oxygen. This is fine because your gut is anaerobic. And they've evolved with humans over millions of years. And just to give some examples of that, so the bacteria in your gut can metabolize carbohydrates, which we cannot, and this creates energy for us. So fiber is a good example of this. A lot of the fiber that we eat we don't have the enzymes to digest it, but a lot of the bacteria in our gut do. And so by metabolizing these carbohydrates, they create energy for themselves, but they also release byproducts, which are energy for us as well. So there's estimates that up to 10% of our daily calorific intake comes from the metabolism of our gut microbes. Another example, and this is one that constantly amazes me, is that breast milk contains sugars that are there to promote growth of specific bacteria and not the baby. So the breast milk of mammals, including humans, they contain certain sugars that are indigestible to the baby and are there to nurture the growth of bacteria in the gut. And not all bacteria, I'm talking here about specific species of bacteria. So this is a great example of how we have evolved with these bacteria. And I'll lead on to it, but it kind of shows how beneficial they are to our health. If you think about that as mammals, we've evolved to produce milk at an energy cost to us that is there to nurture bacteria and not our newborn baby. Okay, so the next thing I was to ask was another poll, and this is what influences your intestinal microbiota. And I've all of these, so birth mode, whether you're vaginally delivered or cesarean section delivered, diet, medications, exercise, antibiotics, or your age, or all of the above? And the answer to this is all of the above. So starting from when you're born, we know that whether you're born, whether you're vaginally delivered or delivered via C-section, you acquire a different microbial community that colonizes your gut. And so for cesarean section babies, so while C-sections are a very essential and life-saving medical intervention, there's going to be people on this call who are born via C-section or have delivered babies via C-section. It is an extremely important intervention, but we know it does affect the microbial community that you acquire in early life. So these communities, they converge over time, so we get the same species. But what we think is happening is that this early period of life is an important window during which the immune system develops. So if you're born via cesarean section, you're picking up bacteria that are on your mother's skin or from the hospital environment, and these are the first bacteria your immune system is interacting with. And so there have been some studies that have shown that C-section born babies have a higher incidence of asthma in later life, and we think this is because of how the immune system is developing over time. But to unpick these, what we need is longitudinal studies. So by this I mean sampling the microbiome of babies when they're born and following them for the first few years of life, and integrating this with health records, and we need big numbers as well. So we're getting there, but I don't quite think we're there yet. And this will feed into later on in my talk when I'm talking about the idea of association versus causation and some of the hype around the microbiome. So the diet you eat plays a big role in shaping your microbiome. So all of these different colors here indicate different bacterial groups. So whether you're breastfed or formula fed, when babies are weaned and go on to solid food, we see a big shift in the composition of the microbiota. So some species disappear and other species come in, and these tend to be the species that represent an adult composition that stay with you for the rest of your life. And then, of course, the medications you're taking, the antibiotics, and how healthy you are all impact as well. And then as you get older, then we start to see a shift in the composition of your microbiome as well. So a good way to think of how your microbiome develops over time, over the first few years of life, and so really I'm talking about the first 5 years of life, is to imagine as an ecological succession. So a lot of the theory that we use in studying the gut microbiome, we adopt from studies of other ecological environments. So you can think of when a baby is born, it's colonized by really quick growing species that can grow in the presence of oxygen that is present in the baby's gut at the start of life. So like E. coli, for example. But over time, these are replaced by more anaerobic species that cannot grow in the presence of oxygen. And then eventually we get to the end point is a climax community where you have hundreds of different species interacting with each other and cooperating and competing with each other. So it's quite interesting to think about the fact that your gut ecosystem, it matures in a similar way to other ecosystems out there. Okay, so how do we study the microbiome? So I'm going to simplify this, but in terms of studying the intestinal microbiota, for in most cases, we start with fecal samples. So these are a good representation of what's in the large intestine. And they're quite easy to get. So what we do is we extract the DNA from the fecal sample, and then we sequence it. And then once we have the sequence, what we do is we want to identify the bacteria that represent each sequence. So one way to do this is to compare the DNA to a database with known bacterial species. So for example, if you had a sequence that looked like this, and then you come to your database and it matched species X, and you know species X is in there. And so what we're doing is we are trying to estimate the abundance of, we're trying to identify which bacterial species are in a particular sample and how abundant they are. So the question I wanted to ask then was, well, what can we learn from microbiome sample? And really, at a high level, what we want to do is we want to compare what's different in the microbiome between healthy and sick people. And the idea is then that bacteria that are enriched in healthy people could be health associated, or bacteria that are enriched in people with a particular illness could be associated with that disease. And then this brings me on then onto the idea of association versus causation. And so just because there's more or less of a particular bacterial species in a healthy versus a sick person, it doesn't necessarily mean it plays a role. And so this is quite a complicated figure, but these are examples of where the intestinal microbiota has been associated with different illnesses. And so I think some of these, we know the microbiome does play a role, but some of these will be associative as well. That the particular bacterial species identified are not necessarily causative for that disease. So this is really to address some of the hype around microbiome studies. But I think we, as the field is developing, we're getting closer to the answers to these. And it's developing by people, it's developing by studies that are conducted with large numbers of samples, so in the thousands or the tens of thousands, and that are conducted over long periods of time and integrated with health records. And so this is being driven by improvements in sequencing technology, but it's also been driven by improvements in other computational analysis, such as like machine learning approaches, where you can start to identify, using these large studies, which bacteria are associated with a particular illness. So a big focus of our lab, the lab I work in, is to try and address this problem. So I've talked about how we can use sequencing to identify a particular bacterial species that's associated with a disease. But one way to actually to drill down into that and to understand its biology and to understand if it's causative for that disease is to use microbiology approaches and bring it back into the lab and study its biology. So up until a few years ago, a big barrier to doing this was having the actual bacteria to experiment with. So some bacteria grow quite readily. They will grow in simple growth media, but others are quite fastidious, need particular nutrients, and can be quite tricky to grow. So we've developed an approach to culture a lot more bacteria in the gut, and this is involved on this involves, again, starting with your microbiota sample with a fecal sample, and then we use two different approaches to grow them on the plate. So what we do is we take the fecal sample and we dilute it down 5 or 6 times, and then we plate this on an agar plate that contains nutrient agar, and then we allow the individual bacterial colonies to grow. Once these colonies are growing on the plate, we pick them and we identify them by sequencing just a small part of their genome, and then we sequence their entire genome. So now we have a whole genome sequence, and we can use this genome sequence to understand the biology of the bacteria. But we also freeze the actual isolates as well in a -80-degree freezer. So now we have the isolates as well to study. And so we use these isolates for experiments in the lab and also for some mouse studies as well to understand the biology. So if you can grow gut bacteria in the lab, you can understand their biology using experimental means. And this is quite powerful because you can link your experimental findings back to the genome and vice versa.
Hilary, we just had a question from John in the chat box saying,
Sure.
I think it was about your previous slide about association versus causation.
Yeah.
He's asked, how much truth is there in the statement of the microbiome is a better predictor of cancer than the patient's genome?
That's a very good question. So I think I don't know. The background to this is, in recent years, there's been a number of very high-profile studies that showed that the microbiome is predictive of response to certain treatments for cancer. So this is, we were working on a study on this a few years ago. So this is what I know about it. So for some cancers, you can use checkpoint inhibitors, which are designed to provoke the immune system into attacking tumors. And what they found was that when they compared, so some people respond to these and some people don't. And we were involved in a study looking at melanoma patients. And what we found is that, and other people have published it as well, is that the microbiome of people who respond is different to people who don't respond. And so there's an idea here that there's particular bacteria that are interacting with the immune system, and these can promote a response to these checkpoint inhibitors. So this is actually a really nice example of where the microbiome, I think, is going to really deliver and come to therapeutics. So there's clinical trials going on right now to identify the bacterial species that can switch somebody from a non-responder to a responder to these drugs. And then if you can profile their microbiome and say, okay, you're a non-responder, and then give them that bacterial species in a capsule, the idea is you can switch them to a responder and they can respond to these drugs. So I think for some cancers, the microbiome will definitely be able to play a role. I think in terms of, if you just compare the microbiome and the human genome, the human genome is probably going to be more predictive, but the microbiome is going to be important for certain cancers anyway. And some microbes are carcinogenic, like E. Colibacter pylori in the stomach as well. So I don't think I really answered your question, but it was kind of a roundabout answer.
Thank you.
Okay. So we use these anaerobic cabinets to work with the gut bacteria in the lab. And so the question was, why do we use these? And the answer is primarily to keep the oxygen away from the bacteria. So how these cabinets work is you put your arms into these sleeves, and they're sealed. And as you can see, there's a pedal down here. You press the blue pedal, that vacuums and it sucks all the air out of the sleeves. And then you press the white pedal and it blows nitrogen into the sleeves. And you do that 3 times, and the idea is it removes all the oxygen in the sleeves. And then you can't see it here, but there's doors behind here. You can open the doors and you can put your arms in, and then you can work with the bacteria inside. So we spend a lot of time in the lab working in these anaerobic cabinets with gut bacteria. So using our culturing approaches, we have built up a large culture collection of different bacteria. You can think of it like a library so we have hundreds of species and thousands of strains. And here I'm showing another phylogenetic tree, and this time it's just bacteria. And the idea is all of these different branches represent different bacterial groups. And we have these isolates frozen in these frozen glycerol stocks here at -80 degrees, and we can use these for experiments as well. And that's a big focus of the lab that I work in. Okay, now I want to talk about some of the diseases associated with the microbiome and one particular cure. And I'm going to focus on one particular pathogen. So you can, in a healthy person, so if you're a person who is relatively young, eating a good diet, not taking lots of medication or antibiotics, you can imagine your microbiome is like this, like a complex rainforest. So you have lots of species diversity, you have lots of competition for nutrients and spaces, so your bacteria in the gut competing with each other. And this makes it difficult for pathogens or bacteria that can make us sick to colonize and cause infection. Now, if you think about it, we're constantly ingesting bacteria in the surfaces we touch and the food we eat, but it's not that often we actually get sick. And this is due in a large part to what's called colonization resistance. So in the gut of a healthy person, because there's lots of species diversity and lots of competition, it's hard for pathogens to come in and to colonize and cause infection. But in some cases, when you take antibiotics, while they might be very effective at getting rid of what's making you sick, there can also be collateral damage to the intestinal microbiome as well. And so here you have a reduced species diversity, you have less competition for nutrients and spaces. space. And this makes it much easier for pathogens to come in and colonize and cause infection. And in this scenario, you have less colonization resistance. So one particular pathogen that is extremely effective at exploiting this is Clostridium difficile. So this was a major problem in hospitals in the UK and around the world up until a few years ago. And so these are TEM images, transmission electron microscopy images. And so this is roughly 3 micrometers long, so 3 millionths of a meter. And this is the vegetative cell of Clostridium difficile. This is what it normally looks like. But what's interesting about C. diff is that it can make these resilient dormant structures called spores. And you can see one here developing in the vegetative cell. So C. diff isn't the only bacterial species that can make spores. Not all species that do make spores are pathogens. In fact, we know from my work that a lot of them are commensal as well, so beneficial bacteria. But once the spore is fully formed, this vegetative cell lyses and the spore is released. And these spores are extremely resilient. They can resist oxygen, disinfectants, and they can remain dormant until they're picked up by a new host. And it's the spores that have made them such a problem to get rid of in hospitals, because you need really strict cleaning regimes to kill the spores. Now, when you ingest the spores of C. diff, so when the spore reaches the small intestine and it recognizes the presence of bile acids in your gut, this is a trigger, and then it germinates and it forms a new vegetative cell, and then it can colonize the gut. So for most people who are healthy, C. diff is not a problem because of colonization resistance. But for people who come into hospital being treated for something else, or who are immunocompromised, they can get infected with C. diff. And antibiotics can be given as a treatment, and 80% of the time this works. And here you can see what a healthy colon looks like. This isn't what it normally looks like. This is after it's been washed out and there's no food or bacteria in here, but you can see it's a lovely pink color. But in 20% of the cases, antibiotics don't work, and you can get recurrent infection. And this is an example of what your colon looks like, an extremely advanced C. diff infection. You have these infectious pustules. So this is a problem because you can't keep giving more antibiotics because the C. diff is resistant to them, and also the antibiotics are keeping your resident intestinal microbiota down. But an alternative treatment is to do a fecal microbiota transplant, or FMT. And this is the idea of you take feces from a healthy person and you give it to the person with C. diff infection, and the bacteria or the microbiome in the fecal sample from the healthy person will outcompete C. diff and drive it out. And so this has been commercialized, and the leaders in this is a company in the States called OpenBiome. And they take fecal samples, they screen them and make sure there's no pathogens in there, and then they sell them to hospitals around the States. And it's extremely effective for C. diff, but it's also been used for a lot of other illnesses as well. So I think, I included this here because people might have heard about a fecal transplant. And it's quite interesting because the idea is obviously disgusting, but it is highly, highly effective at treating C. diff infection. And so it's approved for use by the NHS as well for treating C. diff. But this slide here is just to show, so on the top here, these are all the trials using FMT, and you can see the numbers are increasing rapidly. And then the bottom half of the figure is all the different conditions that it's been treated for. So we have C. diff here is the one with the highest number of trials. So I think FMT will be effective for some of these illnesses, but it won't be effective for all of them. And ultimately, it remains undefined because you don't know what you're putting into the person. Actually, now that I see it, I can see melanoma here. So FMT has been used as a treatment for melanoma as well. And this comes back to what I was talking about with this idea of checkpoint inhibitors. So the idea is that if you give someone an FMT who has melanoma, that there could be particular bacteria in there that interact with the immune system in a certain way where it can target tumors. But actually, a more effective way to do that would be to identify the specific bacteria that have that effect. And so instead of giving someone a fecal transplant, you give somebody a capsule with, 5 or 10 different bacterial species in it, and you know exactly what they are and you know what's going into the person. And so that's what we're trying to do for different illnesses, and it's where the research in the microbiome field is going as well. And so it's this idea of, can we identify good bacteria that can eliminate pathogens from the gut? So it could be eliminating pathogens from the gut, or it could be, as in the case with melanoma, to identify bacteria that can interact with the immune system in a beneficial way. And so in terms of getting rid of pathogens from the gut, so there's different ways the bacteria in your gut can do this to restore colonization resistance. So I talked earlier about how our microbiome can use fiber as an energy source and ferment it, and it produces these metabolites called short chain fatty acids. These are actually an energy source for our epithelial cells, but some of them are also inhibitory to pathogens. And so the bacteria in our gut will also compete with pathogens for nutrients, so for food in the gut. Some of them produce bacteriocins. These are antimicrobial proteins that can be quite specific, that kill other pathogens. And I also mentioned at the start of the talk, bacteriophages. So these are viruses that infect bacteria. And so these are also being used as a particular treatment as well. Okay, and so I'll leave it there. So I'd just like to acknowledge all of my colleagues based at Sanger and also at EBI as well. And these are people who have contributed to the studies I've discussed during this talk. Thank you very much.
Thanks, Hilary. We've already had a couple of questions come in, but if anyone's got more questions for Hilary, either just unmute or stick them in the chat box and we'll pick them out. So there was a second one from John asking, is it really hard culturing bacteria that are super specialized for particular GI environments?
Yes, it's a simple answer to that. So we have managed to culture a lot of them, but actually subsequent studies, including from Alex here, who I've highlighted, have shown that actually there's still a lot of bacterial species in the gut that remain uncultured. And there's different reasons why this is. Some of them are going to be extremely, extremely oxygen sensitive. So when we get our fecal samples, we try and get them into the anaerobic cabinets that I showed you earlier on as quickly as possible. But some of the bacteria can potentially survive for only seconds in an aerobic environment. Some of them as well will have specific nutrient requirements that we're not providing in the lab. And some of them may also require particular metabolites from other bacteria. So sometimes you see two different looking bacterial colonies that you only see them growing together. And so we think this is because they're feeding off each other. And so yeah, so if you're looking at bacteria in the small intestine or versus the large intestine, they're going to have different nutrient requirements there as well. So you will have, depending on the microenvironment in the gut, they're going to have different requirements to get them to grow in the lab.
Awesome. We then had a question from Matt saying, do probiotic drinks actually have any effect on the microbiome? Do they have any health benefits, or do they fall into the sort of hype side of microbiome research?
I'm on the fence with this one. So I suppose for context, a lot of the bacteria that are in probiotics are like lactobacilli or bifidobacterium. And these are bacteria that are quite easy to grow in the lab. And this is why they have been developed for probiotics. And so there is a lot of evidence out there showing that in some cases these bacteria are beneficial. But I think overall, I think all the claims that are made probably don't add up. It's not the sum of its parts. I think how this is going to change is as research into the gut microbiome has increased, we have a much better idea of the bacteria that are in there. So for example, lactobacilli are not a big component of the gut microbiome. In a lot of cases, these probiotics won't colonize, but it's thought that as they pass through the gut, they exert a beneficial effect. I think this is probably true in some cases, but for a lot of other bacteria, they're probably just washed out. So I think the probiotics of the future are going to be based on different bacteria. So bacteria we know that are abundant, and we can grow them now, and we can understand their health benefits as well.
Awesome. We've got a few more questions coming in, which are all really interesting. So linking to the previous question about culturing different bacteria, someone's put that they've heard that some bacterial species are unculturable. Do you know if this is true? And if it is, do you know why?
So yes, it is true. Some of them are. And no, I don't know why. I think for some of them, it's probably because they're extremely oxygen sensitive. So we grow the bacteria on agar plates, which is a solid media. I suspect some bacteria may not like this. They might prefer a more viscous media, or essentially there's something missing in our laboratory environments that these bacteria like.
Do you think in the future maybe scientists will be able to sort of, test out all of the different options of things they might need to see if they could be culturable?
I think so, yes. There is a lot of interest in this, and people are coming up with new and clever ways to isolate and grow bacteria that haven't been cultured yet. Definitely, yeah.
Awesome. I've had a question from Eleanor asking if a capsule of specific bacteria is given to a patient orally, how can you ensure they colonize in the correct part of the gut?
Yeah, it's an excellent question. So in a lot of cases, how if you're using gut bacteria that are adapted to the gut, you're relying on their ability to transit through the intestines, make it through the low pH of the stomach, and colonize the intestinal environment. And if you think about it, because we all share similar bacteria, so the people you live with, you have a more similar microbiome with them than people you don't live with. And this means that you're sharing your microbiome between the people that you live with. So these bacteria are extremely capable of transmitting from one person to another. And so they can survive for long enough in the external aerobic environment until they're picked up by a new host. And producing spores is one way they can do this. So in a lot of cases, we don't really know how they manage to make it through the low pH of the stomach and to colonize, but we know that they do because they're present in lots of different people. And if they couldn't do this, they would go extinct. So in terms of developing a therapeutic, it's a lot of trial and error to see which bacteria can colonize the best.
Awesome. We've had a question asking, has anyone investigated whether there's a three-dimensional aspect to bacterial communities, or do you work with solely 2D cultures?
We work with 2D cultures that are growing on an agar plate, but other people are looking into like biofilms, so bacteria that will stick together and clump together, and this would be more of a 3D environment, and probably a much more a natural environment to what's in the gut as well. And so, for example, there's a lot of interest in this in the oral microbiome, so the bacteria that colonize your teeth and cause cavities. People are using lots of really cool microscopy to image the bacteria in the gut, and what you tend to find is this biofilm assembles in the same particular manner, where some bugs like being on the inside and some are in the middle and some are on the outside. So yeah, it is something people are looking at, but the majority of research is done on 2D on an agar plate because that's how the field is developed, and it's quite easy to do.
Are there any specific sort of challenges to doing like the 3D aspect rather than a 2D one? I presume there are challenges to growing things or looking at things in 3D.
I mean, I think the best way to look at things in 3D is to look at them in their natural environment. So people have done this looking at how bacteria are present in the intestinal environment as well. So by using fluorescent imaging and looking how these bacteria are interacting in the gut, and then you're actually looking at how they come together in a 3D environment in their natural environment. But this is, I mean, it can be quite tricky to do.
I've got a question from Fran actually asking if you identify a completely new bacteria, what do you use as the genome reference?
So this happens quite a lot. So when we're doing your microbiome study and we get all this, the sequence from the fecal sample, and we compare it back to a database, in a lot of cases the species will match and we know exactly what it is, but in some cases they don't. And what we do then is we compare it to the closest thing it will match. So if it doesn't match at 100%, we ask, okay, what does it match to at 90%, for example? And you know, there's quite a defined process for naming a novel bacterial species. It's something I'm doing now at the moment. So if you have a novel species, you can send it to the specific journal and you describe its biology and you can name it. And there's all sorts of rules with what you can and can't name it. You can't name it after yourself, but you can name it after other people. So it's quite interesting. It's something we're doing a little bit of as well.
And we've had one question asking, and this is quite specific, does the ratio of beneficial and pathogenic bacteria change due to microbial translocation in AIDS patients? So that's quite specific, and don't know if you'll know anything about that.
I don't know specifically about AIDS patients, but I would imagine it probably does, given the status of their immune system. So it's going to be different to a healthy person.
Great. If anyone's got any further questions, do feel free to put them in the chat box. That was a whole host of great questions in a row there. So I'll give everyone a second or two to put any extra questions in. Last time that we had you on, you recommended a couple of books that were particularly good on this topic. Would you mind sharing those with this group this evening?
Yeah, I'm trying to remember the names of them now. There was a Martin Blazer book about antibiotics, and the other book was We Contain Multitudes. I can't remember the author's name. Sorry,
I put you on the spot a bit there.
Okay.
But yeah, last time
Ed Yong is the author of that. Yeah.
Awesome. Last time a lot of the students were asking for good reading recommendations that were not in the hype half of microbiome research. So if any of you want to read it any further, then yeah, those are the recommendations we came up with last time.
Yeah, no, they're good and they're quite accessible as well.
Yeah, I think you've put them on the Padlet, haven't you?
Have I? Well done, me. I have.
So yes.
Awesome.
That was great. Thank you.
Doesn't look like there's any more questions. So Hilary, we will let you go for this evening. So thank you so much for joining us and sharing a talk with this group as well. So yeah, we'll give you back your Wednesday evening.
No problem. Thank you. Thanks, everybody.
Thank you.
And then in the rest of this session, we're going to be sort of sharing teaching resources that will be useful teaching this in the classroom. I know we maybe have some STEM ambassadors with us this evening, so again, useful for that as well. Andrea, no problem. As I said, yeah, the rest will be recorded.
Okay, bye-bye.
Thanks, Hilary.
Thanks, Hilary.
Eleanor, yes, this was recorded, so don't worry, you can watch the first half of this. Yeah, the whole thing's been recorded, so you can catch up on anything that you missed. The recording will be available probably by the end of the week. Yep. So let me find my slides.
So where we can put it, we'll put it on our It's Learning platform if you have registered for the It's Learning platform, but you email a link out anyway, don't you, Em?
Yeah, so most of you I think have signed up through Eventbrite, so there'll be like a follow-up Eventbrite email that will have any useful links and the recording, and that'll probably be on Friday. But if not, I'll pop our email address in the chat box at the end as well. So if anyone's got further questions, then they can ask.
Yeah.
So we've got a Padlet for the rest of this session that you should be able to access without a login. So Padlet's kind of like an interactive whiteboard. So the idea is that we've put some bits up, including those books that I forgot I'd added there. And so this is all the resources that we'd like to share with you. And if you've got any that you'd like to share with each other, this is a great place to put them so that anyone that's watching on record can sort of pick them up later as well. So I'll pop the link for that in the chat box. So that should be there. And linked there as well is the learning resource sort of leaflet for this session as well. So all of these learning resources are sort of aimed at around year 10 plus, or about age 14, 15 plus. So sort of selection of reading resources and videos that you or students could use to sort of to learn more about this topic. And then Fran, I'll hand over to you to talk more specifically about this activity.
Brilliant. Yeah, so there was a reason I asked about the genome reference, because I thought that might nicely link into this activity. So this can be used to talk about the microbiome, but it could also be used to talk about antimicrobial resistance as well. So I think if you've been on one of these talks before, you might be familiar with it, but there are some new faces, which is fantastic. So this is called Construct a Bug, and the idea of this is to offer a resource to sort of show students that you can find out about a bacteria, what it is, from looking at its genome sequence. So if we go to the next slide, so every activity we have on Your Genome has got PowerPoint slides with it, and this is one of them. And this slide basically is like, looking down a microscope, it is not always very easy to spot the differences between bacteria, because they can be very minor changes in their genomes that make them actually quite different. They may physically look the same, but they could be genetically different. And obviously when you're looking at the microbiome, it's not always obvious what those bacteria are without looking at the genome. So if we go to the next slide, so Hilary did a lovely slide showing how we do that. But basically, if we want to sequence any bacteria, whether it's from the microbiome or from an environmental sample, we basically have to sequence it. We pop it into things like the MySeq or the HiSeq. We get the DNA sequence, and then we can analyze it using bioinformatic tools and compare it to other different genomes. So again, this slide sort of simply sort of shows those different steps. The picture at the left-hand side shows the sort of MySeq, the HiSeq sequences, and this is our data center here where all the data gets stored and processed. And currently our data center has around 90 petabytes of data. It's absolutely huge due to the number of different programs that we have running. So what's next? There we go. So this again breaks down what those processes are. So again, all of these images and slides are available if you want them, but this breaks down that you sequence it, you need to quality control your sequence to make sure it's good enough for analysis. It then gets overlapped to assemble. This is quite critical because this is what's linked to this activity. We then annotate it so we can find out what its function is, and then we compare to other genomes. So that's essentially how the data is processed, and those are the main key steps in the process. So what our activity involves, there's several resources which you can print out. So you've got the sequence read. So the idea is that you print these out, and you then have to cut them, cut all the different sequences out. You can see there's lots of different fragment sizes. And the idea is that you then would randomize these. You could either put them in a bag and students pull out a few, and then they have to match them to the reference genomes. So this is representing perhaps a microbiome sample. You've got a real mixture of DNA in there, and then you have to match it to the sort of similar species using these reference guides here. And then the next step is you've got gene profiles, so you can actually see what genes have been annotated and what if they have sequence in those genes. You can then have the worksheet that will get them to sort of ask some key questions. So, you know, what features or adaptations does it have, or could it have? Is it pathogenic? Is it not pathogenic? You know, you can see which is it most closely related to. We then also have bacterial profiles. So on here at the top, so you've got like Klebsiella. I don't know why I'm pointing at my screen because you can't actually see what I'm pointing at. Schwannella, which is quite an interesting one, and Salmonella typhi. So this is quite pathogenic. So we could do another version that's a bit more microbiome, but these, again, some of these can be found. And of course, we've also got PowerPoint slides to go with it. So what's the next slide in? So basically, like I said, you get your fragments, the students work in groups, they maybe have 5 fragments each, then they have to match them up to the different genes here. So you've got one there, which is KPC2, or you've got LUXS, so there. And then what they can do is see what that is most similar to Klebsiella. And this is essentially what Hilary does. They get these fragments, they see what is it most similar to, and that is how you can sort of either identify new species or similar species or exact matching species when you look at the genome analysis. So that's the first bit there. And then you can construct it. You can then find out, well, what are the genes that are present in these bacteria? So is it toxin defense? Is it community genes that now enable them to do a bit of quorum sensing, so they know how many are in there? That is you quite see a lot of bacteria that are sort of in the gut have a lot of this, seeing how full is the environment, can they spread out further, for example. And then the third bit is you basically look at your reference and work out what it's most related to. So this is all literally downloadable. You can download them from the Your Genome website. So again, it focuses on your adaptation, survival strategies, relatedness to other species, and the gene function, and whether things have been conserved, because generally if genes have been conserved, they perform a very vital function as well. So these are all sort of key things that you can talk to around the microbiome, but also around sort of bacterial adaptations and survival as well. So there we go. So that's that one activity. So I don't know if that would be of interest to you. Like I said, it is all ready to go on Your Genome. If there's any, we can use this forum. You can come, like, speak on unmute if you want. But if there's any other activities that you're aware of that you would like to share with the rest of the group, we always use this opportunity as a way of sharing practice as well. So I don't know if you've got any ideas for other activities that you think would link quite nicely. Or this is a completely new area and we need to find out some more stuff for it.
I'd say, like, I did sort of a bit of research before these sessions as to other activities, and there's really doesn't seem to be quite as much out there for sort of secondary microbiome. There's quite a bit for primary, but it doesn't seem to be that many activities around the microbiome at secondary. I don't know if that's just it's not a topic that's taught as much, or I don't know.
So in your curriculum, so question to the group, in your curriculums that you currently teach, is there a direct reference to microbiomes, or is there sort of more of an enrichment around sort of talking about communal species and general bacteria, or is it the focus very much on pathogenic bacteria? Again, you can unmute or you can put your suggestions in the chat. Oh, flurry of stuff.
Chipping in. Can you hear me?
Yeah,
oh, yeah,
I can hear you, Eleanor.
Oh yeah, I'm trainee teacher at the moment, so I'm kind of acquiring kind of ideas, but there's kind of communicable disease element of the teaching, so it covers the sort of bacteria. I can see this type of activity would kind of cross-relate with the sort of evolution and survival of the fittest, so actually it would be quite a good sort of linking activity between sort of communicable diseases element of the curriculum and the sort of inheritance evolution side of things. So I mean, that's where I would possibly kind of link it to two different elements.
Yeah, no, that's a really good point, actually. Yeah, and it is, and it's, the thing with bacteria is they evolve very quickly and they adapt. They acquire changes in their DNA. So this is how all the different resistance occur, or you know, they evolve very quickly. So this is a really nice linker. Yeah, completely agree, Eleanor.
Matt's put something sort of similar, that on 8QA there's no specific mention of microbiome, but I'd like to try and discuss it at some point. So yeah, I think it sort of skirts around the curriculum rather than actually being named itself. And John's got an idea for making the activity better. So is it possible to have some pre-made random sequences as chopping them up and randomizing them sucked? Is that
I'm guessing he's done the activity and he did not enjoy cutting up the little bits of paper.
Is that for this bit, John? For this sheet? Okay.
No. And you said your line manager struggled with a year 11 group. So was it the content or was it the top line?
You're okay.
Yeah. So yeah, so we need to find a way of or just having less fiddly bits to cut out. I think that might be a good top tip, because who wants to spend their evening cutting up bits of paper?
I just had a quick look in the I just had a quick look in my AQA GCSE textbook. And the other area where it might fit into is sort of like the fighting disease, and how a microbiome and healthy microbiome can actually counteract pathogenic bacteria. And I don't know if there's any sort of resources that you could do on that. I'm just thinking how you could fit kind of this more novel research into the existing curriculum as it sits in the textbook. That actually, you know, yeah, the sort of elements of introducing sort of, you know, probiotics or the kind of boosting the immune system within the gut by encouraging certain, you know, bacteria in the microbiome is actually protective against pathogenic bacteria, but in a way that's sort of nice and easy and obvious that to digest for, you know, year 10s and 11s.
Yeah, no, that's a really good idea. Like I say, we always like to use these meets just to sort of to find out what people want and how we can improve our offer. So that's really useful. Cool. I can't even pronounce that. Mephanobacter smithii is a cool extremophile example. Yeah, that sounds like a very cool bacteria. Fantastic. Is that the ones that are found in volcanoes? I'm guessing with the methane thing, that could be that.
Yeah, it's not one I've heard of, but it's in the gut. There we go.
Yeah.
We've all learned something new this evening.
There you go. John's always full of little gems. Cool.
Awesome.
Brilliant.
Well, if anyone else does try out this activity and like John thinks some of the instructions need tweaking, just let us know. We obviously want to make sure that anything we make is as usable as possible for sort of anyone. So I will pop our email in the chat box if I can spell it.
Okay. I was going to say there are some new names on the board today, which is fantastic. So if you're not already a member of the Bioscience Lite community and would like a login to our It's Learning platform, which is an online platform where we've got lots of other resources for teaching these topics and so on, please do email us and we can add you on here. And it's a really great place to find new ideas and share your ideas. There's announcements, so you find out when the next talks are coming up, and you can have access to all the recordings as well. So if we find out think there's any new, really cool stuff that you might be interested in, we put those in the announcements as well, saying, well, there's a really good article on this. So it's a nice community to use. If you are already on there and not using it, you know, maybe a little reminder to use it, but we realize you're very busy. So so definitely recommend that as well. And it's all basically a very supportive teaching community. Can you put the Padlet links on It's Learning?
Yes, will do.
Yes, definitely. So
I'll put the recording of this and the Padlet links both on the Eventbrite email and on It's Learning, so that you hopefully can't miss them.
Yeah,
they'll be everywhere.
Awesome, brilliant. And I said, if you've got any ideas for the Padlet board, feel, you know, again, it's there we want to use it as a sharing best practice community. So by all means, add your own ideas. If you've come across anything that you like or find interesting, then we definitely recommend you use that as well. So any other questions or anything we can help with while we're here? Otherwise, we can finish up a little early.
I'll give everyone a chance to put something in the chat box if you've got any questions, but I'll just let you know about our next session. So our next topic is evolution and protein. So we've got a speaker called Zachary Arden, who researches how evolution creates new proteins, things like orphan genes and hypothetical proteins that I'd never heard of. So I'm really looking forward to his talk. The student-based talk, so anyone that's got upper secondary students, is on the 24th of February. And then our teacher session, just like this, will be on Wednesday the 9th of March. So I will find the sign up link for that because I've got it somewhere. But again, that'll also be on the Eventbrite email, and it will be on the It's Learning platform as well. So really looking forward to that. I think it'll be a really interesting talk.
Brilliant. Well, thanks for everyone for coming along. It's been lovely to have you here and find out more about microbiomes. Like I said, hopefully we'll see you at another Bioscience Lite next month. But have a good evening, everyone. Em's just put the link in the chat for you, so you can see the next link to that.
Awesome. Thank you, everyone.
Thank you.